All right, welcome back, everyone. Today, I'm going to do something a little bit different. I'm going to, instead of making a PowerPoint presentation, I'm going to take you through section 3.2, which is talking about cell organelles, and I'm going to take you through it on visible body because I think it's something that you'll be able to use in your further science courses and your science careers. And also, it's something that you're getting and you can use in this class too. So it might help just to figure out what we're doing with it. So, um, in the last lecture, we talked about the cell membrane and how it's a selectively permeable membrane that allows certain things in and it lets certain things out. But let's take a look at what it's holding inside. So before we jump into this, let me just point out that there are two types of cells, and all living things are made up of two types of cells, either prokaryotic cells or eukaryotic cells. So the bacteria and archaea are comprised of prokaryotic cells. That's right here, the prokaryotic cell. Um, in fact, some of those organisms are single-celled organisms, and probably most of them are an organism all to themselves that is a single cell. Uh, the rest of life on Earth, animals, plants, fungi, etc., are comprised of these eukaryotic cells. And let's take a look at some of the difference. So the first different part is the size. Look, this is zero point 0.1 to 10 micrometers. That's that little mu, which looks like a U with a little tail on the left side. That's called mu, and it just means micrometers, so micrometers. And this is 10 to 100, so 10 times at least as big. Um, first, the first major difference is that eukaryotic cells have a nucleus, which contains the DNA, whereas prokaryotic cells do not have a nucleus. They just have a place in the middle of the cell that contains the DNA that we call the nucleoid region, but it doesn't have its own membrane, whereas a nucleus has its own membrane. We'll talk more about what a nucleus is in another lecture, but they both have ribosomes. Ribosomes are the organelle that produces proteins by joining amino acids together. They both have cytoplasm. They both have a plasma membrane. But the rest of the differences come in terms of the organelles, these membrane-bound organelles. Eukaryotic cells have these membrane-bound organelles that we're going to talk about today, whereas prokaryotic cells do not. Okay, let's get rid of that. So if I was to come in here to visible body and I wanted to look at something that's in the cell, I'm going to go to click on cell structure and function, and if I did that, it would bring up this image. This is a 3D model of what a eukaryotic cell might look like, and it's really interesting because you can click on certain parts of it, it'll highlight it for you, and you can even click and scroll and look around this 3D model. It's really interesting, and I think it's going to be great to use for this lecture, especially instead of, I don't know, just looking at static pictures on the screen. Um, so the first thing I want to talk about is that cells are filled with this jelly-like substance, and it holds all their components, and it also provides this fluid-like substance or medium for their reactions to take place. This is called the cytosol. So take a look at the cell. I'm going to click on cytosol, and you can see that it sort of highlights where the cytosol would be. And if so, this is what we're looking at is like if I took a knife and I cut like all of the cell membrane out, and so I could look into the cell, this is what we would see. So you can notice the cytosol sort of would fill this whole area, but we've taken that part out. So the cytosol is just like the jelly-like substance that would be holding things in the cell. Let's take a look at the organelles. Let's get rid of let's hide the cytosol so you can see the rest of the organelles that were hidden in that jelly-like substance. And let's click on organelles. So organelles is this big term for everything basically that's left in this cell. Those are your organelles. The word organelle just means little organ. What they are is they are membrane-bound which means they have membranes themselves, so they're protected by the cell membrane, but these also have their own membranes. And they each perform their own specific functions, and we'll look at all of them today. But when you take the organelles and the cytosol together, that's called the cytoplasm. So it's just like everything that's inside of a cell is called the cytoplasm. All right, let's first start with the cytoskeleton. Okay, the cytoskeleton, it's just like your skeleton. It's just like your skeleton supports your body, the cytoskeleton supports the cell. It doesn't just support the cell structurally, it'll also play a role in cell movement, cell reproduction, and the transport of materials within the cell. So the cytoskeleton is going to be made up of 3 different types of filaments. The largest of these filaments are the microtubules. So here they are, and they're coming out of this organelle that we'll talk about in just a second. But microtubules are comprised of subunits called tubulin. So this is like a protein called tubulin. They give the cell the shape and also comprise the cilia and flagella. And if you know what cilia and flagella are, maybe from previous lectures, they help cells move. So once again, they're the largest of the filaments. They're made up of subunits called tubulin, and they give the cell the shape, and they also can help the cell move. So this organelle right here is called the centrosome. See, it's highlighted now. It was in yellow, now it's in this green color. That's the centrosome, and it's the origin point for microtubules. It builds microtubules. Inside, it has 2 what are called centrioles. The centrioles, your book says that they are what produces the microtubules, and you know, sometimes that is what books use, but really the entire centrosome is what I like to think of, and most biologists will think of as what produces the microtubules, and what centrioles actually do is still kind of up in the air, I believe. So let's just say that the centrosome is the organelle that produces the microtubules. It contains 2 centrioles. Okay. The second type of filament is the microfilament. So let's click on that. Here are the microfilaments. They're the smallest of the filaments, and it's made up of this protein called actin. Microfilaments constitute a large component of your muscle tissue. Actin, along with another protein, myosin, are responsible for muscle contraction. Also, actin filaments will help a cell to divide during cell division. They create the cleavage furrow that'll eventually split the cell into 2. So once we talk about cell replication, you'll see what a cleavage furrow looks like, and that happens due to these microfilaments. The last filament is the intermediate filament. These actually aren't pictured on visible body, but just like their name might imply, they are the in-between size of microtubules and microfilaments. And these guys are important for maintaining cell structure as well, but instead of making sure that the cell doesn't get squished, they resist tension that might normally rip the cells apart. So you need these microtubules, microfilaments, intermediate filaments for the cell to have its structure. All right, I think we're good here. Let's move to the next one. And the next organelle is the endoplasmic reticulum. So the endoplasmic reticulum, let's click it, is this entire grand structure here, okay? And the ER is what they often call the endoplasmic reticulum, so you don't have to say that long word. The ER is the system of channels that's continuous with the nuclear membrane. So when we talk about the nucleus here in a second, I've clicked it. It's actually hidden by. Let's see if we can hide. Let's see, hide. Yes, here you go. So here's the nucleus, and it has its own nuclear membrane or nuclear envelope, which means that it has its own membrane, right? All these organelles have its own membrane. But when we bring back, here you go. When we bring back the endoplasmic reticulum, it is continuous with that nuclear membrane. So it's attached right to it. The endoplasmic reticulum can exist in 2 forms. The first, let's click on it, is the rough ER. So let's see if I can hide the smooth ER. Here's the rough ER. I want to hide you. Okay, here's the rough ER, and let's see if I zoom in. you'll see that the rough ER has these little balls on it, these little studs on it, and those are another type of organelle called ribosomes. Ribosomes are the organelle, like I said before, that serves as a site of protein synthesis. So it might be unsurprising to you to know that the primary job of the rough ER is for the synthesis and modification of proteins. But these ribosomes also are found in the cytoplasm in general, in the cytosol in general. So the ribosomes that are attached to the rough ER make proteins that are going to be destined to either go be part of the cell membrane or to leave the cell in general. And so what happens is the rough ER will synthesize and modify proteins that will then be packaged up and exported from the cell. Let's bring back the smooth ER. So the smooth ER, if you look closely here, there's no ribosomes attached to it. That one's just sort of floating in the cytoplasm. But they have no studded ribosomes. So the smooth ER, its function is to produce lipids. Okay, so just a small difference there. The rough ER produces proteins. The smooth ER produces lipids. I think we're good with that. We can move on to the next organelle, which is the Golgi apparatus, and they call it the Golgi complex here. But here, I'll click it. That's this one right here. The Golgi apparatus is sort of like the post office of the cell. It's responsible for sorting, modifying, and eventually shipping off products that come from the rough ER. The Golgi apparatus kind of looks like these stacked, flattened discs, almost like oddly shaped pancakes. Like the ER, these discs are membranous, so they have their own membrane. The Golgi apparatus has two distinct sides, right? So there's like an entrance and an exit. Here's the rough ER, so the product will come in from this side, go and be modified in the Golgi apparatus, and exit through this side. So they're going to be released in vesicles, in little pieces of membrane that are holding the materials. And if the product that it has is supposed to be exported from the cell, the vesicle will migrate to the cell surface, the cell membrane. The vesicle then will fuse to the cell, and the cargo will be secreted. So remember when we talked about exocytosis, that's what that is. Okay, I think we can move on from that. The next that they want us to talk about are lysosomes. Let's click on those and figure out where they are. Okay, so they're here. Lysosomes are over here. And peroxisomes are very, very similar, so those are going to be over here. They're pretty much identical looking, but their processes are a little bit different. A lysosome, once again, those are over here, is an organelle that contains enzymes that break down and digest unneeded cellular components, like damaged organelles or things in general that need to be gotten rid of. And that is right in the name. Lysosomes, lys means to cut. And so similar organelles are like peroxisomes. They are there to neutralize toxins. And if you look at the name perox, that name suggests exactly what they do. They'll produce hydrogen peroxide, which is H2O2, which is not toxic to cells, and they produce that from things that are toxic to cells. And so they're kind of the same idea. They get rid of things that the cell needs to get rid of, but they just do them in slightly different ways. Let's go to the next one, which is the nucleus. And we're going to get into this with Dr. Droud, his lecture on chapter 3.3. But I'll just tell you really quickly, let's look at the nucleus. So the nucleus contains the cell's genetic information, which is encoded in molecules of DNA, and those molecules of DNA are sort of wrapped up and bundled into things called chromosomes. Those are the chromosomes right there. If you are a human, you get two copies of each chromosome, one from your mom and one from your dad. Overall, we have 23 pairs, or 46 in total chromosomes as a human. The nucleus is separated from the cytosol by its own membrane, the nuclear envelope. And inside the nucleus, there's this central body called the nucleolus or nucleol. I guess I just call it nucleolus, or some people call it nucleolus. But it produces the ribosomes. And like I said, those ribosomes are the organelles that make proteins. Interestingly enough, what you'll hear, DNA has to be copied into RNA, which then is copied into proteins. So all of the really important stuff happens in the nucleus. It's sort of the brain center of the cell, the control center of the cell. Now let's move on to the next one. And here's sort of like the famous one that everyone kind of knows when you're taking biology for the first time in high school. Mitochondria, right? Like the powerhouse of the cell. Notice that the mitochondria, they have both a, let's see if I can get it to show up. They have a outer membrane. If I hide that, they also have an inner membrane. And maybe if we talk about the production of ATP, we'll talk about why that's important. But this is a double membraned organelle. It has an inner membrane, it has an outer membrane. But mitochondria, they use oxygen to do aerobic respiration. If you remember the process of aerobic respiration, that whole process is to produce ATP, or the cell's energy dollar, right? That's what I usually like to call it. And those ATP molecules are the cell's fuel, the cell's energy. Last here is just showing you the ribosomes. There are both free ribosomes and the attached ribosomes on the rough ER. So I think with that, we're going to call it a day and look forward to more coming very soon.